Okay, I lied before. We're actually doing this on page 77, and we are going to be talking about nitrogenous bases. So let's title this Nitrogenous Bases. I'm going to put a background around this thing. Okay, nitrogenous bases. There are two different nitrogenous bases. There are your big ones and your little ones, or your large ones and your small ones. And there are two large ones and two small ones. There's your A's, your T's, your C's, and your G's. And we are going to start with the small ones, which are made up of one ring. So I'm going to draw that here. Now this ring is not a sugar. It has nitrogen in it. So it's not carbon, hydrogen, oxygen like glucose or ribose or lactose or whatever. It has nitrogen. I'm not going to draw all of that because it's not that important. But what you should know is that this ring has nitrogen, hence nitrogenous base. Now the ones that are made up of one ring are your thymine and your cytosine. And so that's going to be your T's and your C's. Okay, there are two more bases. They are both large, and we are going to draw those as well. Now, the first ring, oops, that's ugly, looks a lot like this other one. It's six-sided. We're good to go. But there's another ring, and it is a five-sided ring. And that makes this ring physically bigger than this ring. Now, the ones that are large are your A's or your adenines and your G's or your guanines. So A and G. And here's how I remember which ones are large and which ones are small. The word large has an A and a G in it. So those are large. The small ones are cute. That has a C and a T in it. So the little ones are cute, C and T. The large ones are A and G. All right, now these fit together in order to make that whole DNA structure fit together. So we've got this where we've got the sugars and then the nitrogen bases are pulling those things together. So we've got one nitrogenous base on this side and one nitrogenous base on this side, and those connect in the middle, which is going to hold that sugar phosphate backbone in place. So these have to somehow fit together, and there's a very specific way that they fit together, and we're going to draw that. So I'm going to start by drawing a T. Now T is a small one. It's always going to fit with a big one. And the big one that it always is going to fit with is adenine. So I'm going to draw my adenine over here. So I'm going to start with this six-sided, and then I'm going to add my five-sided ring. Now, my A's and my T's are held together in the center of the structure, and they are held together by something called hydrogen bonds. Now, we've talked about bonds before, but most of the bonds that we've talked about are called covalent bonds, and covalent bonds are much stronger than hydrogen bonds. Now, my A's and my T's are held together by two hydrogen bonds. Next, I'm going to show you how my C's and my G's fit together. So if A always binds with T, then C always binds with G. Now I could put my C here and my G here, and then I would have the small ones on one side and the big ones on the other side. And it can happen that way, but it can also happen where it alternates. Or maybe you end up with 3 small ones and then a big one. And that's totally fine. Now, when I put the big one over here, the part that actually connects to the sugar phosphate backbone over here is the 5-sided part. So what that means is I'm actually going to stagger this so that my 6-sided part is over here. And then my 5-sided part is here. And that's going to make it so that my backbone can run right here along this. So this is going to be a G, and G bonds with C. So I'm going to draw C. And G bonds to C with 3 hydrogen bonds. And hydrogen bonds are pretty weak, which means that they can actually break apart, which will allow the strands to actually separate. Now let's make a note on here so that we can remember how this works. So first of all, A binds to T. With 2 hydrogen bonds. And then C, binds to G with 3 hydrogen bonds. And this is going to be something that you are going to want to remember. So an easy way to remember it, one of my students a few years ago came up with this, is there are 2 apples in the tree. So 2 hydrogen bonds between A and T, apple tree. If you're fancy, you might have a house that has a 3-car garage. So there are 3 hydrogen bonds between C and G. So 2 apples in the tree, 3-car garage. And that's just a good way to remember how these fit together. T and A only bind with each other. C and G only bind with each other. All right, the last thing that we're going to put on this page is that DNA is a polymer. So we've talked about polymers before, and DNA is arguably the most important polymer for life. And polymers are many things stuck together. The monomer of DNA is called a nucleotide. So I'm going to draw a whole nucleotide here so that it's easy for us to see what a nucleotide looks like. So I'm going to start with my phosphate. So I've got an OH, P double bonded to O, O, O minus. That's my phosphate. Now my phosphate is bonded to my sugar. And you're going to be so good at drawing these by the end of this class. So there's my sugar. And then my sugar is connected to, and it's connected off of the first carbon, the sugar is connected to that nitrogenous base. So I'm going to actually color these a little bit so that we can see what they are. I'm going to shade my whole phosphate. It's going to be red. that's my phosphate. Next, I'm going to have my deoxyribose sugar, and then my nitrogenous base. But it was my phosphate that I labeled red. Let's label this sugar blue. Blue on blue can be a little bit hard to read, but you get the idea. And then last but not least, my nitrogenous base, I'm going to make green because that is the colored pencil that I have available. You can color it however you want. Now this whole thing, sugar, phosphate, and nitrogenous base, is called a nucleotide. So DNA is made up of a nucleotide bonded to a nucleotide, bonded to a nucleotide, bonded to a nucleotide. The nucleotides are the monomers, DNA is the polymer. So we're going to switch back to that model. So go ahead and get those cut up pieces ready, and then we're going to finish off that model.